Okay, so I'd like to today, like I usually do, start out reviewing what we did last time. And then I've got a little more stuff to talk about in terms of cancer. And hopefully, you noticed last night I posted some questions that we could go over. So I'd rather not forge ahead and do the next material right now because I feel like we've done a lot already and we still have 4 more weeks. So let's do this today. And then next week, I'm going to start in on a whole new section. It's about signal transduction in early development. Um, so I don't want to get too much into embryo development because that's not what the class is about. But what I really like about this material is it shows how different hormonal influences cause changes in differentiation that are really important early on here in development. So all these signal transduction pathways we've talked about already are like vital in early development. So we'll start that on Monday. So today, let us review what we did last time first. So where is my share? There it is. Sharing my screen. Share. Okay. So now you're seeing my screen, and here's where we left off. So last week we talked about this, the changes in cells as they go from normal to more and more messed up and then eventually invasive. Um, and skip over this. We did go over this a couple of times already. Okay, changes in cancer cells. So the first thing that we talk about here is how cancer cells are not fully differentiated. We say de-differentiation, but basically sometimes they can go backwards in differentiation, or sometimes they just never mature. And that's the case of immune cells. So here's just examples of what we mean by differentiation. It has to do with the history of growth factors that cells are exposed to that send them down different pathways. Um, so again, not just one hormonal influence, but this hormone turns on these genes. Some of them are transcription factors. Now the next hormone then can change that response, but it depends on what came beforehand. So that's differentiation. So and what we're talking about with cancer cells is they're not differentiating. Here's normal skin tissue, um, kind of rounded cells at the bottom that um, are stem cells that are dividing. As they divide, one cell moves up and differentiates, and over time it goes further and further differentiated till it's basically just a sac of keratin, and then it sloughs off. And in de-differentiation, these cells don't differentiate on the way up, and then also they just divide more and they become more and more screwed up, having to do with DNA damage. Basically, we talk about that later in this chapter. So de-differentiation. The other is two things. You've got anchorage independent growth and loss of density dependent inhibition. So in terms of anchorage dependent growth, actually we learned both of these through in vitro things where you took cells and you put them on a microscope slide and they went into apoptosis, and people realized later you need to put collagen down on that slide in order for the cells to survive. And that's all about activating the focal adhesions that turn on signal transduction pathways that provide survival signals. And this involves blocking a lot of apoptotic signals. BAD especially is kind of key to this. BAD is a pro-apoptotic BCL2, and both AKT and MAP kinase, or ERK, are going to phosphorylate BAD, and that's going to stop apoptosis from happening. So, so basically, normally when cells are not connected to the extracellular matrix, they're going to go into apoptosis. And here goes a fire truck by here. And oh God, it's distracting me now. Where was I going with this? Oh, so normally they have to be connected to collagen. So cancer cells have to find a way to inhibit apoptosis and activate their survival signals without the focal adhesions attaching. Also, you've got your cadherins, and cadherins are telling the cell that they're attached to other cells, and so they should stop dividing. You know, the tissue's full now. Don't make more cells because we're all packed together already. They lose this signal transduction here as well, and they continue to divide, and that's what forms a tumor that's going to like expand into the tissue and bulge out because they are no longer inhibited by that density inhibition. And again, this was learned in vitro. They learned that you put cells in culture, and normal cells, you give them growth factors and everything, they will grow till they reach confluence. They're all touching each other and they stop dividing. So cancer cells get around that. Telomerase, just skip to the end here. The whole idea with telomerase is with your linear chromosome, every time you divide, you'll lose a little bit of information at the 2 5' ends, and the chromosome gets shorter and shorter. And telomerase is going to come in and add telomerase is going to come in and add base pairs on the end by having its own internal template, and it base pairs with the end of the chromosome, puts down new DNA, and leaves the same thing on the end here, so then it can come up and around and then reanneal and then add more DNA. So you get these tandem repeats at the end of the chromosome, and all it's doing is just extending that chromosome so that it'll be safe in further rounds of replication. So telomerase is the one people think of in terms of aging. You know, cells get old and cells die eventually because they don't have telomerase. Craziest thing, you know, I showed you this. I like to tell my stories. I'm an Irishman. I told you this before. I showed you this before. When I put this animation up online, people started finding it through Google, and all these people would email me and ask, where can I get telomerase? Are there telomerase pills available for me so I can live longer? And it's like, you don't really want to activate your telomerase because that's going to cause your cells to become cancerous. So, and besides, if you take a pill with a protein in it, it's going to be digested anyway. So anyway, telomerase, making the cells immortal. And then this part, this is kind of central to all these other changes, really, because it's signal transduction that's controlling all the other things. But basically, your cancer cells are going to need self-sufficient growth signals, which means they don't need to be stimulated from externally with growth factors. They've already got the internal mechanisms turned on. And these internal mechanisms are going to do things like activate telomerase, and they're going to take the place of all of this. So they take care of this as well, and they overcome this. So really, this self-sufficient growth signal is really causing all the other changes we see in cancer cells. So when we talk about self-sufficient growth signals, three avenues you got to look at. One is activating the pro-growth regulators, and those are any enzyme in the pathway between hormone, or even including the hormone, all the way through activating DEGs and passage through the restriction point. So anything that's pushing you forward on that pathway is going to be a pro-growth regulator, and they're going to become activated by cancer cells. Inhibition of tumor suppressors, so like GSK3 beta would be a tumor suppressor. As long as you've got it there, you're going to be blocking beta-catenin. But if you lose GSK3 beta, beta-catenin builds up. Same thing with APC, Axin, WTX, any of these. If they're not present, the destruction complex won't work, and the cell will divide based on beta-catenin activation. Other tumor suppressors, things like CDKIs, right? If you lose those, then the cell moves forward when it shouldn't. RB being a central tumor suppressor, blocking E2F. And again, remember, when you're activating cell division, that means you're turning on E2F. And if you overexpress E2F, that activates ARF, alternate reading frame, which then causes the cell to go into apoptosis. So at the same time you're activating all these pro-growth things, you need to inhibit apoptosis, because otherwise the cell will just die. So we'll talk later about the two-hit hypothesis, or the multi-hit hypothesis, which is that one mutation alone is not going to make the cell cancerous or definitely not metastatic. You need several things to change for that cell to become really dangerous, because we need to activate these pathways and block these pathways. Okay, angiogenesis. I like angiogenesis. The main thing I like about it is it gives us a really good target to get rid of tumors, because a tumor needs a blood supply in order to grow. And so, when the tumor becomes hypoxic in the center, oxygen can only diffuse so far, and the inner cells of this cell mass are going to become hypoxic, and they'll start releasing growth factors to recruit a blood supply to it. Things like VEGF, vascular endothelial growth factor, and FGF. So these are going to be released. Checking my notes to make sure I didn't forget anything. Yep, VEGF and FGF. And some cells can also release inhibitors like angiostatin or endostatin. So that, again, there's always a balance between pro and anti-growth things going on. Here, your tumor is favoring the pro-growth regulators, releasing VEGF, and that's causing these endothelial cells to divide and start to become mobile, and then move into the tumor and bring it a blood supply. And here are some of the really awesome new drugs that have been produced in the last 20 years. Again, I don't think I said this before, but typically your old school chemotherapy agents are metabolic poisons. They're things that are going to mess up or block DNA replication, or mitotic spindle inhibitors, all these drugs that are going to cause dividing cells to grow. And the problem with that is, and this is where all the side effects come in, because there are a lot of cells that do need to keep dividing normally, right? All your stem cells in your GI tract, your hair cells are constantly growing, your immune cells are constantly dividing and replacing dead cells. So when you hit someone with these classic chemotherapy agents, you're causing all this damage all over the body, and you're just hoping that you're going to preferentially kill those cancer cells because they're dividing faster than everything else. But it's pretty dramatic what those old school things do. Here's the new line of chemotherapy, which is really amazing, which is something like Avastin. It's an antibody that binds VEGF, so that blocks the angiogenesis, and they've had really good success with that. Here's another one doing the same thing. And then we've got specific kinase inhibitors that are going to block the cell signaling here, to block the self-sufficient growth signals and things. So new drugs on the market thanks to cell biologists. Okay, so now we're talking about cells that are starting to move. What allows them to move? And it comes down to letting go, increasing motility, and clearing a path in front of the cells. So letting go is all about our focal adhesions and adherens junctions. As I said before, the focal adhesion signals that it's attached to extracellular matrix, and also the cell can signal the integrin to let go of extracellular matrix. So when the cell starts to become mobile, it will let go, and then let go and reform these as the cell starts to crawl. And it lets go of its neighboring cells that allow it that allows it to leave the tumor and start crawling into other areas. And crawling happens because of all the mechanisms we talked about in chapter 3 with cell movement. RAC-RO CEC42 are going to remodel the cytoskeleton, put it into a crawling mode, and so we've activated these G proteins, and then the cell starts to crawl, which involves reaching out, letting go back here, reaching out, grabbing up here, pulling the cell forward into that, and letting go behind. Increased motility. And then we've got clearing a path. So your cells are totally coated in extracellular matrix proteins like collagen, fibronectin, and that's holding them in place. And also we've got a lot of protein here between the epithelial cells and the stromal layer that's right underneath the epithelial cells. That's our basal lamina. And that is the key to the difference between normal and malignant normal. A cell that's dividing too much and one that's actually malignant is when it has the ability to cross that basal lamina. So what cancer cells will do is release tissue plasminogen activator or plasminogen activator. And plasminogen activator will activate plasmin. Plasmin will then activate our matrix metalloproteinases. So these are proteases that are on the extracellular matrix already in a pro-enzyme form, so they're turned off. Plasmin will turn them on, and then you break down collagen, and now the cell's got like a soupy mix it can swim through on its way into the body. So clear a path. All right, now what? Oh, causes of cancer. So I said on Monday that there are some virally caused cancers. They're not that common, but there are a few virally caused cancers. One is HPV, and HPV will infect a cell, and it could just stay in kind of the lysogenic cycle and stay dormant for a long time. Or like in this example, it replicates and releases virins, but doesn't kill the cell. And then over time, it can progress to a malignant cancer cell. So here we have invasive cancer. So that's the HPV, and then we also have the Epstein-Barr virus. Or is this still HPV? No, this is still HPV. So what I like about this is it shows how this virus gets around the normal mechanisms to inhibit cell division. It's got two proteins, E7 and E6. E7 inhibits RB, releasing E2F. But again, E2F will cause apoptosis in addition to cell division. But E6 then is going to block P53, and P53 is involved in that P14 and apoptosis mechanism. So you're blocking apoptosis and activating cell division, and that allows the cells to become cancerous. Again, blocking both pathways. And then Epstein-Barr is different in that it actually produces two receptors that are very similar to receptors that are normally in these cells. And let's give us a little shadow here. These things are going to activate signal transduction pathways. They're going to inhibit apoptosis. And so everything that a normal growth stimulating mutation would do are being done by these receptors that are being expressed in the membrane. Okay, so the other thing that's going to cause cancer is activation of pro-growth genes. So I talked about this a little bit earlier, just today. But basically, anything along the pathway between the growth factor and the DEGs, if it's out of control, is going to be an oncogene. So all these things are normally in our genome, but they're proto-oncogenes because they're under control. Once the cell loses control of them, they're oncogenes. And this could be anything from a growth factor, which is being released and causing autocrine stimulation, growth factor receptor, RAS, G proteins, kinases, transcription factors, IEGs and DEGs. Oh, here's our DEGs down here. These are IEGs. So our pro-growth regulators there. So any of these that have become out of control, they can be carcinogenic. And the question now is, how do they get out of control? What makes these things get out of control? And that is DNA mutations. So we like to think of cancer as a disease of DNA DNA damage. Excuse me. So these are 5 different ways that DNA can become damaged that turns a proto-oncogene into an oncogene. And I don't know about you, but whenever I think about mutations, this is usually where my mind goes, point mutations. So you're just changing one or a few bases, and that can change some amino acids in the cell. I'm sorry, in the protein, and that can cause it to become constitutively active. Good example of that is RAS. RAS is a protein that's got 3 kind of vulnerable amino acids that if you change any one of those, it doesn't interact with GAP anymore, and so it's constitutively turned on. It can't break down GTP anymore, so it's always on, and that makes it oncogenic. It's a classic example of a point mutation. Here's another point mutation, can cause loss of an exon or something. This is the N terminal of the protein. So somehow we've skipped over a big chunk of the protein, and now we don't have any extracellular domain, and these become constitutively active. So this is a receptor tyrosine kinase that is just the transmembrane domain and the kinase domain, and that's constitutively active. I believe that's what TRAC is, or ERB. ERB, I think, is the one that is activated that way. Okay. Overexpression. Now here's another great way to mess up a cell. Not mutating the protein, just having the normal protein, but expressing too much or at the wrong time in the cell, and that can cause a proto-oncogene to become oncogenic. One way you can overexpress is by gene amplification, and that happens during DNA replication. The DNA is so plastic, it does so many strange things, and normally your cell's correcting that, but sometimes it doesn't. So you can get a normal gene just duplicated over and over again until you've got multiple copies of it, and then you get a lot more of the protein. If you've got 3 genes instead of 1, then you've got 3 times as much DNA being made, as much protein being made, I'm sorry. And again, with these receptor tyrosine kinases, if you overexpress them, they activate themselves in the absence of growth factor. Another way to get overexpression is by translocation, taking your proto-oncogene that's normally in a quiet chromosome and putting it into an active chromosome. And basically swapping a chunk of one chromosome for a chunk of another chromosome. And it's a really nice model of how that happens. Here's one chromosome, here is the other one. So, and again, we're not talking homologous chromosomes. We're talking totally different chromosomes. Take a chunk off this one, chunk off that one, swap them, and now you've got 2 new chromosomes. So this is what they call heterologous recombination. I believe that's what it's called. It's not homologous recombination, which is, chromosome 11 swapping with chromosome 11, but, chromosome 11 with 15, that's heterologous recombination, and it is dangerous. Here's why it's dangerous. So again, this is overexpression. You take MYC from a chromosome 8, where normally in these lymphocytes, they're fully mature, they've gone through their differentiation, and this part of chromosome 8 is all condensed down, so the promoter is not accessible from MYC. And in chapter 4 chromosome 14 is where we're making our immunoglobulin heavy chains, which are very heavily transcribed in immune cells. And so this is a very active region of this chromosome. So now we're taking this chunk and swapping with that chunk, and now we've got MYC next to a very active region. The promoter is accessible. We start making MYC ectopically, sort of, where it's not supposed to be, and that causes this chromosome, Burkitt's lymphoma. And then here's an example of chromosomal translocation that results in a whole new protein. So this is the ABL and the BCR proteins, which chromosome 9 swaps with chromosome 11, makes a brand new chromosome, which is the Philadelphia chromosome. And in the middle of it, we have a brand new protein. So we're taking BCR, and we're taking this much of BCR, and we're sticking it on the front of ABL. And we have BCR ABL now, and this is unregulated. So you've got a kinase that's just unregulated, so self-division growth signals. And, this is not curable, really. I guess the only way to cure this would be to do a total bone marrow destruction and then reseed with new bone marrow, because it's genetic at this point. These cells are just going to be there with this screwed up chromosome. And here's what the protein looks like, and here's some really awesome drugs. Again, selective drugs, not killing all cells, but just stopping these cells from dividing. And then local DNA rearrangements. And here we can have an insertion, which is going to mess up a protein. It could make it hyperactive, could just destroy its function. You have to knock out tumor suppressors as well. So this could just destroy a tumor suppressor. You could delete a chunk out of a thing and destroy a tumor suppressor. Or you could delete a piece of a protein that allows it to be active. A lot of our proteins are activated by cleaving off a self-inhibitory portion of the protein. Cleave that off, and now it's constitutively active. And then here, inversion or transposition, where kind of a gene, it seems as though it jumps out, flips around, and jumps back in. And it has to do, again, with that DNA replication mechanism. And this is what it kind of looks like. Breaks here and here, flips around, and reconnects. Well, here we're actually taking this out, taking this big chunk of DNA out. Here's the inversion, breaking here and here and flipping over and reinserting. And here's this protein, which I just think this mechanism is so cool. It's not cool if you have it, obviously, because it's cancer causing, but I like the way this mechanism works. So you've got tropomyosin here, tropomyosin being a coiled coil protein, and NTRK, which is the NGF receptor. Cut it here, flip it around, and now you've got the growth factor receptor with tropomyosin on it. So you get a coiled coil, and this is constitutively active now because it's always going to be bound to itself. So that's local chromosome rearrangements. Retroviral insertion. So, this can happen naturally with retroviruses, or, where a virus will insert itself into the DNA. And the viral DNA is going to be really active. But if it inserts itself right next to something that's supposed to be quiet, then this becomes active. Just like that MYC example we just talked about. Put a viral DNA next to MYC, and now you're getting a lot of MYC. Here's your retroviral lifespan here. The virus enters the cell, releases its RNA. From the RNA, it makes reverse transcriptase to make the DNA. DNA inserts into the genome, and again, if it's right next to an important gene, it could turn that gene on. Okay, so I don't think I did tumor suppressors, did I? I don't think so. So that's it for the review. I'm sorry?
I was going to say, I just said no.
Okay, good. Thank you. So increase my volume here. I keep losing my mouse. There it is. Okay, so all these DNA mutations are going to lead to either activation of pro-growth regulators, turning a proto-oncogene into an oncogene, or knocking out our tumor suppressors. So what are tumor suppressors? We've got two classes of tumor suppressors. One are the gatekeepers. That's usually what we think about in terms of tumor suppressors. And then we also have caretakers. So the gatekeepers are ones that are controlling cell cycle, and caretakers are the ones that are involved in DNA repair. So gatekeepers being APC, which is inhibit Wnt signaling inhibits GSK3 beta. APC is part of that GSK3 beta complex. RB and P53, PI3, P10. You remember P10 is the enzyme that takes phosphate off PIP3, which is going to turn off everything that's got a pH domain. RB, classic binding E2F. SMAD4, which is part of our TGF beta pathway. This is the cellular SMAD that binds with whatever class of receptor SMAD is being activated. We'll talk about that when we get into cell development too, how there's different classes of receptor SMADs. But anyway, TGF beta is typically inhibiting cell division, as we saw last week. So knocking that out is pro-growth regulating. And P53, which is controlling for DNA damage. As we said, 50% of all cancers have P53 knocked out, which causes more and more mutations to occur. And then we've got these guys down here. You've probably heard of BRCA1 and BRCA2. These are involved in double-strand repair breaks, mismatch repair, DNA excision, XPA, XPB. Excision repair has to do, a lot of excision repair has to do with thymine dimers, which you get from UV radiation, cutting those sections out and then replacing them with the correct bases. So this is from an XPA deletion, xeroderma pigmentosum. This kid can never see the sunlight, basically, because he has no way of repairing the thymine dimers that occur when UV light hits his skin. Okay, so those are our tumor suppressors. Again, remember, gatekeepers, caretakers. Some are protecting, is blocking cell cycle. You could put your CDK inhibitors into this as well. And then your caretakers are the ones that are making sure your DNA is correct. Okay, so how do these mutations happen? What's happening? One thing is carcinogens, and there are a lot of carcinogens. I'm emphasizing smoking here because a lot of our undergraduates still smoke, if you can believe it. God, when I was in high school, you knew this is like in the 70s. It's like, oh, I don't want to start smoking. Smoking causes cancer. Here we are, 40 years later, people are still smoking cigarettes. I don't get it. But anyway, carcinogens. carcinogens are things that are going to mutate DNA. And you can see this is, it's so clear. This is as cigarette consumption goes up, delayed behind it, you start seeing cancer deaths. And then smoking goes down, delayed behind that, cancer deaths start coming down. So the correlation, of course, but the causation is there as well. The more cigarettes you smoke per day, the higher your chance of dying. The earlier you start smoking, the higher your chance of dying. And after you quit smoking, your odds of dying are going down. So stop smoking your damn cigarettes is what I'm saying. Marijuana too, although people don't smoke 25 joints a day, right? But anytime you burn something and inhale it, you're producing these polyaromatic hydrocarbons, and they're cancer causing. Also happens when you barbecue, right? When you have the whole idea of the barbecue is you've got your coals there that are not all that efficient in burning their carbon, so you're getting these just black soot all over everything. And oh, does it taste good. Love me a good charred steak. Okay, but it's also cancer causing. Here's an interesting factoid about your liver. Your liver, I've talked about it before, the P450 system is there to break down toxins. But again, it's not looking, it couldn't be there to look for specific toxins, right? It's like whatever you happen to eat, the liver's going to take that. And if it's something it doesn't recognize, it's going to change it chemically. And the whole idea is to try and make that chemical either inactive or more water soluble so your kidneys can get rid of it. Most of the time, your liver's inactivating things. Sometimes it activates them, though. Psilocybin, one of my favorite examples. Psilocybin is not active biologically until it goes to your liver and gets converted into a psilocin, which is active. But anyway, getting back to this figure, basically, bacteria require histidine to grow unless they're mutated. So here we're saying if you take these bacteria and you put the substance in with those bacteria, very few of them will develop the ability to grow without histidine. Okay, so it takes a mutation to be able to make their own histidine. So you just your substance on these bacteria, and some of them will mutate, but not many. You take that substance, you incubate it with a liver homogenate, and let the cytochrome P450s work on it. Now add it to those bacteria, and quite a few mutations happen, so you get a lot more colonies. So the liver is actually activating this substance rather than inhibiting it. So there are a lot of pre-carcinogens that can get converted by your liver into carcinogens, which is why I had my liver removed, because I don't want this to happen to me, so I don't have a liver. How else can we produce mutations? One is radiation, either ionizing or non-ionizing radiation. Here is your spectrum of light, right, from radio, microwave, infrared, visible. When you get to the higher end, there's more energy in it. The ultraviolet sunlight then can cause thymine dimers to happen. Basically, you activate two thymines and they bind to each other, and they're no longer binding to their A's over here, and that makes a kink in the DNA. And that poor kid earlier, if he had XPA would recognize this and cut at either end and then repair that DNA. But ultraviolet then causes these thymine dimers. And then your higher energy radiation is actually just going to break the DNA. And that's when you start getting, chromosomal translocations and other kinds of large changes in the DNA that can activate your proto-oncogenes. In terms of carcinogens, we've got two different types. You've got initiators and promoters. So your initiator, like DMBA, you only need to be exposed to once. and that's going to mutate DNA. It's going to cause point mutations or whatever, cause some kind of mutation in DNA. But that itself is not sometimes going to cause cancer. Some other things like croton oil, which has got 4-ball esters in it. 4-ball esters activate protein kinase C. Croton oil itself is not going to cause cancers to happen either. But if you pre-expose with an initiator and then give the promoter, then you can get tumors to start forming and dividing. I believe in your notes I mentioned androgens can be a promoter as well. So, you hear sometimes with guys with like, I think prostate cancer is one of these where one of the things they do is they do testicular God, I can't even say it. That's how upset it makes me. Amputation, testicular amputation, because your testicles are putting out testosterone, and the testosterone is a promoter for that prostate cancer. So the only way to get rid of it is that. And they also have drugs like Depo-Provera that are going to block that, but not a great way to go, because I mean, loss of testosterone, you get tired, you lose muscle mass. It's just difficult. But it's that or dying. I guess you do that. But anyway, stepwise development of cancer. So this is what I was talking about earlier with how knocking out one protein or activating one protein typically doesn't make a cancer really, really dangerous. You need multiple hits for it to happen. And you can see that in this experiment where you take a mouse that's got some, you give it melanoma, okay? You inject it with melanoma cells, give it melanoma, which is going to metastasize pretty quickly. So some of these melanoma cells make it to the lungs. You take one of those tumors out of the lungs from the new melanoma, you inject that, and you get even more metastases. So now, after having gone through one mouse, more mutations have happened, and it's become more aggressive. So the second mouse, you get even more metastases. You take one of these tumors out, you inject it into a third mouse, and you get even more metastases. So repeating this cycle of removing and injecting is kind of allowing those cancers to become more and more aggressive, which means, developing more and more DNA mutations. Once you start knocking out the caretakers, then, DNA changes can happen a lot easier, and repeated rounds causes more DNA damage and more aggressive tumors. Here, this is another one I tend to harp on a lot because I really like this. This is your large intestine, your colon. Colon cells at the surface are being replaced constantly by cells in these crypts. So that you've got stem cells at the bottom of the crypt that divide, and then cells move up this way and differentiate as they go. And this is a Wnt pathway is causing the division down here. So as you go up here, Wnt signaling stops, they differentiate, and they move up onto the surface, and eventually they're going to die up here and slough off and constantly being replaced. So you're constantly replacing that. And what can happen is down here in the crypt, you get, say, let me zoom in on this. You may get a tumor that takes out APC, okay? Take out APC, beta-catenin builds up. Now these cells are growing too fast, and they're growing faster than they can move up, so they start to fill in the lumen of the crypt here. You then get later, these cells, or probably just one of these cells, gets another mutation, and you get a constitutively active RAS. Now it's growing even more out of control. You're getting even more of these cells filling up that crypt. And then you get another mutation later on. And again, it could be just one of these cells develops one of these mutations, knocks out P53, knocks out BAX. These are growth inhibitors. These are apoptosis activators, activating PI3 kinase, inhibiting PTEN. Basically, more and more mutations start to happen. Starts to go through that whole process of letting go, crawling and clearing out a path, and they become invasive. So bottom line, what I'm trying to say here is it's not just one mutation that causes cancer, but one mutation makes the cells somewhat abnormal, and one of those cells will then become more abnormal, and it'll come to predominate in the tumor, and one of the cells in that will then mutate again and become more predominant, and you that's your progression. Here's our APC again. If you remember, the normal APC is part of the destruction complex where we're breaking down beta-catenin. When Wnt's around, like at the bottom of that crypt, we activate beta-catenin and we divide. If you lose APC, then you no longer have any way to block beta-catenin from building up. And even in the absence of Wnt, we're getting beta-catenin building up and the cells dividing. What was I going to say about that? APC. Oh, so that's why it's called APC, Edematous polyposis coli. This protein was discovered in colon cancer because it's commonly mutated in colon cancer. Okay. And now we're into our review, hallmarks of cancer. Self-sufficiency of growth signals, right? And that's really the key, because at that point, you're going to activate all these other things. Insensitivity to anti-growth signals like TGF-beta and activating SMAD4. Evasion of apoptosis, got to block those pathways. Limitless replicative potential, which is you're activating telomerase, so the cells can continue to divide. Sustained angiogenesis, so you can get yourself a blood supply. And finally, tissue invasion and metastasis, releasing proteases to move through the cell, move through the tissues. Okay, let's stop sharing now. Sharing is caring. Okay, how was that? I think I went a little bit fast, but should have been okay. Any questions about all of that stuff? Everybody okay? Do you want to go over these questions from? Let me show you. So these are the questions I posted in Brightspace. Did you guys get a chance to look at these? Brittany?
I got to look at it for about 30 minutes before class.
Yeah.
Okay. Why don't I give you another couple days to look at that, and we'll talk about it Monday.
Okay.
Because it's better if you struggle through it a bit before I give you the answers than if I just give you the answers. So let's wait, hold off on that. Okay?
Sounds good.
Sounds good.
Okay. So we'll have a quiz this weekend. I'll put it together this afternoon, have it up for you tomorrow. And Monday we will go over these questions. Let me stop sharing them. Monday we'll go over these questions, and then we'll start a little bit of signal transduction and developmental biology, which I think is a lot of fun. I really enjoy it. So any questions or comments?
Nope.
We're all good? All right, guys. Have a great weekend. Study cell biology. Yay.
See ya.
Thank you too.
Bye, Dr. Slitch.